Welcome to What You Don't Know You Don't Know, the podcast that dives headfirst into the surprising, the overlooked, and the misunderstood. Hosted by a curious duo in one man, one woman, we explore the world's most fascinating ideas hiding in plain sight. From science and religion to politics, psychology, history, and everything in between, we shine a light on the knowledge that's publicly available but rarely discussed. Each episode is a deep but never dull conversation that challenges assumptions, sparks new perspectives, and reveals just how much more there is to know, especially about the things you didn't even know you were missing. Whether you're a skeptic, a seeker, or just someone who loves good conversation, this show will leave you thinking, questioning, and most importantly, knowing.
Today, we're going to take a deep dive into a scientific enigma that's, well, it's persisted for nearly 50 years now.
Right, the Viking missions.
Exactly. The unexpected, and let's be honest, kind of controversial, results from the life detection experiments they carried back in 1976.
It was such a landmark moment. Viking 1 and 2, they weren't just taking pictures. Their core mission was really astrobiology.
Right.
They landed on Mars with this whole suite of instruments designed specifically to sample the Martian soil, test it for signs of life, and one experiment, well, one in particular, gave results that have just fueled debate ever since.
Yeah. So our mission, in this deep dive is to try and cut through all those decades of discussion. We're going to unpack the original Viking findings. Look at how, 40 plus years of new discoveries on Mars stuff Viking just couldn't tell us, have forced scientists to completely reevaluate those initial results.
It's a whole new context now.
Exactly, and figure out what this lingering mystery means for the search for life on Mars right now. We've got a stack of articles, research papers, reviews right here, and we're going to distill the really important nuggets for you from this material.
Think of it as getting the essential insights, right? The crucial details from the actual experiments, the pivotal discoveries later on that changed everything, and the core arguments that, keep this question alive.
Absolutely. So settle in, because you're joining us on this deep dive through the data, straight to the core of one of Mars's most persistent scientific questions.
Okay, let's set the scene. 1976. The Viking landers touched down. They're ready to run their biology experiments. There are three main ones: the gas exchange, or GX.
GX, right.
The pyrolytic release, or PR, and the one that's really the heart of our mystery today, the labeled relief, or LR, experiment.
And the LR, it sounds simple on the surface, but it was pretty ingeniously designed, wasn't it? To look for a really fundamental characteristic of life, metabolism.
Exactly. The concept was, well, quite elegant, really. You take a sample of Martian soil, you add a nutrient solution, think of it like microbial food.
Okay.
That contained organic compounds tagged with a radioactive carbon isotope, carbon-14.
Right, the label.
Yeah. And the hypothesis was, look, if there are active microbes in the soil, they'll consume these nutrients, they'll metabolize them, and in doing that, they'll release gas containing that radioactive carbon.
So you detect the radioactive gas.
You detected metabolism,
Yeah.
a signature of life.
Okay. Seems straightforward enough. So what did Viking find when it ran this LR experiment?
Well, the results were positive.
Positive.
Yeah, on both Viking 1 and Viking 2. And these landing sites were separated by over 4,000 miles. When the radioactive nutrient solution was added to the soil, radioactive gas was released. So for Dr. Gilbert Levin, who was the principal investigator, and his team, this looked remarkably like the kind of microbial respiration the experiment was designed to find.
Okay, hold on. Positive result, 2 different sites, thousands of miles apart. That sounds pretty definitive. Why wasn't that the end of the story? Why weren't the headlines everywhere shouting life found on Mars?
Well, because Viking carried another crucial instrument, and its results seemed to completely contradict the LR findings.
Okay.
This was the gas chromatograph mass spectrometer, the GCMS experiment. Its job was different. It was looking for the organic molecules themselves. The carbon-based building blocks that life, at least life as we know it on Earth, is made from.
The ingredients for life. And the GCMS found?
Essentially nothing. It reported that organic molecules were below its detection limit.
Nothing.
Basically, yeah. And this was the major roadblock for saying the LR results meant life. The scientific logic at the time was, well, pretty simple. No organics means no building blocks. And if there are no building blocks, how can you possibly have life carrying out metabolism?
So, okay, even though one experiment got a signal that looked exactly like life, another experiment didn't find the basic ingredients for that life, and the scientific consensus just sided with the lack of ingredients.
Precisely. That was the conclusion.
Yeah.
The scientific community decided that the positive LR signal, despite how it looked, must be due to some kind of non-biological chemical reaction happening in the Martian soil.
Some weird chemistry.
Yeah, they just didn't know exactly what chemistry yet. But Dr. Gilbert-Levin, he never accepted this conclusion. He's argued for the biological interpretation ever since, pointing to very specific details in the LR data.
And for decades, that was pretty much where things stood, right? A curious anomaly, chalked up to chemistry, end of story for most people.
Until
much, much later.
Correct. And this is where new discoveries start to dramatically shift the perspective.
The plot twist, you could say. What happened?
Okay, fast forward now to 2008. The Phoenix lander touches down near Mars's North Pole, makes a really pivotal discovery. Perchlorate. It's present in the Martian soil.
Perchlorate.
Yes. And this is a reactive salt, and crucially, it behaves interestingly when you heat it.
Okay, perchlorate. So how does finding this chemical, like over 30 years later, change how we understand a 1976 experiment?
Well, what's fascinating here is how it potentially explains the GCMS's negative result for organics. Remember, the GCMS worked by heating the soil samples to analyze them.
Right, heat was involved.
Yeah, and subsequent research done much later showed that if you heat Martian soil that contains perchlorate, the perchlorate can decompose. And in that process, it can actually destroy any organic compounds that might have been there in the first place.
Wait a minute, hang on. So the instrument designed to find the organics might have been accidentally destroying them as it looked? A false negative? That's huge.
It really is. It completely changes how you interpret that key piece of counter-evidence that seemed to rule out life. There was a 2010 study by Rafael Navarro-Gonzalez and his team that tested this directly. They took soil from Chile's Atacama Desert. It's organic rich, shares some traits with Mars soil.
A Mars analog?
Exactly. They added magnesium perchlorate, heated it up just like Viking did.
Yeah.
And not only were the organics destroyed, but they detected chlorinated organic molecules as a byproduct.
And Viking's GCMS did find chlorinated organic molecules, but didn't they dismiss them as contamination from Earth?
They did, exactly. This study provided really compelling evidence that maybe those chlorinated compounds weren't contamination at all. They were likely created right there on Mars, inside the GCMS instrument itself, when the perchlorate reacted with native Martian organics during the heating process.
So the implication is just profound. Mars soil might actually contain organic compounds, maybe at levels of a few parts per million, kind of like dry desert soils on Earth.
Right.
So the original big reason to dismiss the LR results the apparent lack of building blocks that's now significantly weakened.
It really removes a major scientific hurdle to even considering the possibility of Martian life. And interestingly, the perchlorate discovery might also tie back to the LR results themselves, though it's a bit more complex there.
How so? Could the perchlorate, or maybe something it breaks down into, actually cause that positive LR signal chemically?
Possibly. Some research, like a paper by McKay, Quinn, and Stoker, suggests that hypochlorite, which can form when cosmic rays interact with perchlorate, could be a reactive chemical agent. And that could potentially mimic the LR response when you add a nutrient solution.
So it's offered up as one potential non-biological explanation for the positive signal itself.
Okay, so perchlorate is doing double duty here. It potentially explains the GCMS puzzle and offers a possible chemical explanation for the LR signal, though in different ways.
Right. But this raises a really important question, though. Even with perchlorate in the mix, if there are plausible chemical explanations, how strong are those original arguments for biology from the LR data itself? What did Levin and his colleagues see that made them stick to their guns for so long?
Yeah, that's where we really need to dig into the specific behaviors of the LR response in the original data, especially the results from the control experiments.
Controls, right.
Reviews like the 2016 paper by Gilbert-Levin and Patricia Ann Stratt, they highlight several key points that they argue are just very difficult for simple chemistry to explain.
Okay, lay those out for us. What details in the LR data does the biological argument really lean on most heavily?
Okay, first, the positive response, the gas release, was completely eliminated by heating the soil sample to a high temperature, 160 degrees Celsius, for 3 hours.
Sterilization, basically.
Exactly. Consistent with killing biological agents, like sterilizing a sample.
Okay, but like you mentioned, high heat can break down some chemicals too. What's the stronger point then?
Well, the second point is often cited as maybe the strongest piece of evidence for biology. The response was significantly reduced by a much lower heat treatment, only 50 degrees Celsius for 3 hours.
50 degrees C, that's not very hot.
No. And this partial inactivation at relatively mild temperatures is, well, it's highly characteristic of biological systems. Most simple chemical oxidants or reactants, they aren't drastically affected by such a low temperature. But many microbes, especially if they're adapted to very cold environments like Mars, would be damaged or killed by even that mild heat.
That is a compelling distinction. Okay, what else? What else did they see?
Third thing, the activity in the samples just disappeared after they were stored for about 2 months. In the dark, at the sort of ambient temperatures inside the lander, which were around 10 to 26 degrees Celsius, the signal just faded away over time.
It just stopped reacting.
Yeah, which is consistent with organisms dying off, maybe due to lack of nutrients or just environmental stress over time. But it's not typical behavior for a stable chemical compound, which you'd expect to just sit there and remain reactive pretty much indefinitely.
So the sample basically went inert over time, just sitting there. That really does sound less like chemistry just waiting around to react.
Exactly. And fourth, the actual shape and the intensity of the responses, how quickly the gas was released, how much gas came out, those kinetics were very similar to responses seen in terrestrial soils known to contain microbes, including soils tested in lab setups trying to mimic Mars conditions.
And this wasn't just a fluke at one spot.
Correct. Similar results showing all these characteristics were obtained at both landing sites, those two widely separated locations, which suggests whatever this active agent was, it was widespread on Mars. So Levin and Stratt, in their review, they argue that while various chemical hypotheses have been proposed over the years, none has been definitively shown to replicate all of these specific characteristics of the LR data simultaneously, especially that sensitivity to 50-degree heat and the inactivation during long-term storage. Those are the tough ones for chemistry to match.
So, let me see if I've got this right. Well, perchlorate gives us a potential cause for a chemical reaction, maybe this hypochlorite thing.
Right.
The specific behavior of the LRR response, particularly those thermal controls and the storage decay, still doesn't fit neatly with the simple chemical models proposed so far.
That's really the core of the biological argument as it stands today.
Right.
When we evaluate the non-biological explanations, that perchlorate, hypochlorite idea, like the one from Quinn and others, it's considered maybe the most promising chemical candidate so far. Because yes, hypochlorite can be broken down by high heat, the 160 degrees C, which matches that specific control.
But, and this is a big but, you're saying it doesn't fully explain everything else.
Not yet, at least based on the published research we're looking at. The sources point out it hasn't been demonstrated that hypochlorite activity would be significantly reduced by only 50 degrees C heating, or that it would completely disappear after just a couple of months of storage at those moderate temperatures. Those crucial controls from the original Viking data are still proving hard for the purely chemical models to replicate all at once.
And what about those earlier chemical ideas, the ones proposed even before perchlorate was found? You mentioned a study using Atacama soil and something called formate.
Right, yeah. Navarro-Gonzalez also did an earlier study back in 2003 where they got a reaction from Atacama soil using labeled formate that looked, well, a bit like the LR signal. But, and this is what the Levin and Stratt review really highlights, they used a vastly higher concentration of formate, like 2,000 times what Viking used.
Wow.
And crucially, they didn't run the high heat sterilization control on that specific formate reaction. And reactions involving formate, they're known to occur non-biologically and aren't always eliminated by high heat, unlike the definitive Viking result. So while it was interesting lab work, that specific study didn't really replicate the key Viking controls. And most of the other earlier general oxidant theories also kind of fell short on explaining the thermal controls convincingly.
Okay, so the picture emerging is really complex. Viking gets a signal that looks like biology, but initially seems to lack the necessary ingredients. Then perchlorate comes along, potentially solves the ingredients problem, the GCMS false negative.
Right, opens that door again.
But the proposed chemical explanation based on perchlorate, this hypochlorite idea, still struggles to match all the observed behaviors of the LR signal, particularly that low heat sensitivity and the storage decay.
You've captured the core tension perfectly. That's exactly where things stand with interpreting the Viking data itself. And then, to add another layer to this whole thing, we now know so much more about the actual Martian environment than we did back in 1976, thanks to all the missions after Viking.
Right. So our whole view of Mars itself has changed.
Significantly, yeah. Back then, Mars was largely seen as, bone-dry, geologically dead, static. Now, we have compelling evidence of abundant water in its past.
For sure.
And even indications of low amounts of liquid water and brines existing today. Maybe transiently, maybe seasonally, but along with widespread ice and absorbed water in the soil.
Things like the findings at Gale Crater.
Exactly. Evidence for transient liquid water activity there. It suggests that while water is definitely scarce, its presence, even if limited, does make the environment potentially more favorable for extant life than we previously thought. This overall picture of Mars is just generally more supportive of the possibility of life, which lends, slightly more weight to a biological interpretation of LR than seemed feasible back in '76.
And we now know for sure there are organics there.
Confirmed. Multiple missions. Curiosity, Perseverance. While it's not proof of life in itself, the confirmed presence of complex organic molecules removes another one of those major original reasons to doubt the LR result. The building blocks are present, Viking just couldn't see them properly.
And the methane, that keeps popping up too.
Another intriguing piece of the puzzle, absolutely. On Earth, a significant chunk of methane comes from biological sources, methanogens. Now, Mars methane could certainly have geological origins,
Sure.
but its sporadic detection keeps that door open for a possible biological source in the current Martian environment.
And Earth itself teaches us about life surviving in really harsh extremes, right?
Absolutely. Extremophiles here on Earth thrive in conditions that, we once thought were completely impossible for life. Halophiles, for example, adapted to high salt environments. They could potentially cope with some of the briny Martian water possibilities. And interestingly, there are even microbes on Earth that utilize perchlorate as an energy source. So while high perchlorate levels might make Mars generally uninhabitable for most known terrestrial life, the existence of these extremophiles means we can't rule out adaptation quite so easily.
So all these environmental discoveries, they don't prove Viking found life.
No, not at all.
But they chip away at the original reasons scientists dismissed that possibility as biologically implausible back then.
Exactly. They removed some of the biggest obstacles that made a biological explanation seem so unlikely in 1976. The environment looks, slightly less hostile, and the building blocks are there.
So, okay, when we zoom out, look at the full picture, decades later, what does this deep dive tell us about the Viking results today? Where do we stand?
Well, the core ambiguity, it just persists. The Viking LR experiment produced this signal that, based on its thermal controls, its storage controls, behaves in ways really consistent with biology. But that initial failure to detect organics, which we now think might be explained by perchlorate, and the discovery of perchlorate itself, with its chemical reactivity providing plausible non-biological explanations, it leaves us stuck.
Stuck in ambiguity.
Pretty much. The critical point, highlighted in the sources, is that the proposed chemical explanations haven't yet been shown to fully replicate all the specific behavioral characteristics of the LR data. Particularly, again, the effects of that 50-degree C heat and the long-term storage decay.
So the conclusion from the sources we looked at is still that the Viking results remain inconclusive. We just don't know for sure.
Precisely. That's the bottom line. The Viking results are still ambiguous. They're still subject to interpretation. Neither a biological nor a purely chemical explanation has been definitively proven or disproven as the cause for those LR responses based on all the data we currently have.
And there's a unique significance to this specific data set, isn't there? Something about it being potentially special, untouched.
Yes, that's a really important point the sources emphasize. The Viking LR experiment might be the only data we have from a sample of Martian soil that was potentially biologically pristine when it was tested.
Because later missions weren't sterilized the same way.
Exactly. Subsequent missions haven't been sterilized to the same rigorous, almost extreme degree as Viking was. That was done specifically to avoid contaminating Mars with Earth microbes before we even looked for Martian ones.
Right.
So this makes fully understanding the Viking results, ambiguous as they are, even more critical. It might be our only glimpse ever at the potential activity of a completely untouched Martian sample.
Wow. That's a heavy thought. Our one chance, maybe, at a pristine sample, and we still aren't sure what it told us nearly 50 years later. So how does this ambiguity, this decades-long mystery, inform the search for life on Mars moving forward? What do we do now?
Well, it absolutely underscores the need for more sophisticated, more varied life detection instruments on future missions. Viking taught us a hard lesson about how easily results can be ambiguous and how clever chemistry can potentially mimic biology.
So what kind of experiments do the sources suggest we need? What's next?
Several key things come up. We definitely need much more sensitive searches for organic molecules, crucially using methods that don't rely on high heating, to avoid destroying them the way the Viking GCMS likely did.
Makes sense.
We need further metabolic experiments, maybe building on the LR concept but using different approaches or broader suites of potential nutrients, maybe looking for different waste products.
Okay.
And a very powerful approach that's often mentioned is searching for chiral preferences.
The handedness of molecules.
Exactly. Life on Earth shows a distinct preference for specific handedness left to right of molecules like amino acids and sugars. Non-biological chemistry typically produces a roughly equal mix. Finding a strong chiral preference would be very strong evidence for life.
Yeah, that sounds like a really powerful potential biosignature, hard to explain chemically.
It would be. And also, simpler methods perhaps, like just high-resolution microscopic examination of soil samples, looking for structures that might resemble cells or fossils.
And beyond the pure science of discovery, there are practical implications too, right? For future human missions, sample return.
Definitely. The possibility, however remote it might seem to some, that the Viking LR experiment did detect viable, maybe even just dormant, life. Well, it raises really important questions for astronaut safety and planetary protection.
Can't just assume the soil is sterile.
Exactly. If we send humans, or especially if we bring samples back to Earth, we need to have the most robust understanding possible of what might actually be in that soil. The lingering ambiguity of Viking reinforces the need for extremely stringent protocols for sterilization, containment, and sample handling.
This deep dive has been just a fascinating journey, really, through how scientific understanding evolves. Seeing how new discoveries can completely change the way we look back at old data. The Viking mystery, it isn't neatly solved.
Not by a long shot.
But we see it through such a richer, more complex lens now, thanks to everything we've learned about Mars since 1976.
It's a powerful example, isn't it, of how science isn't just about finding one simple yes or no answer. It's this continuous process of questioning and revisiting old data and refining our understanding based on new evidence. The fact that this debate is still so active after nearly 50 years is, frankly, quite remarkable.
And the thought, just the possibility that those Viking landers way back in '76 might have actually touched down on soil containing Martian life, even if we didn't fully recognize it or couldn't prove it at the time, that's truly provocative.
Absolutely. And that original, potentially pristine data set holds such a unique place in the history of exploration. It really demands our continued attention and scrutiny.
Well, as we wrap up this deep dive, thank you for joining us on this exploration of the Viking legacy and its modern reevaluation. It really leaves us, and hopefully you, with a profound question to consider. Given the potential for chemistry to mimic life's signals so closely, and the very real possibility that life elsewhere might operate in ways we don't yet fully understand or anticipate, how can we be absolutely sure we've found extraterrestrial life definitively when that day finally comes?
Well, that's a wrap. Hope you liked the episode. Don't miss out on new episodes of You Don't Know, You Don't Know. New episodes every week. Like and subscribe.